Yeah, thanks for the lovely introduction, Allison. Can you hear me? There was some issues with my microphone before, but everything's still working. Awesome. Um, right, yeah, I'll just go ahead and share my screen in the interest of time. And I should say, please interrupt me as I'm going. Talking to my screen for a solid hour is not as fun as interacting. So please do interrupt me in the chat or otherwise. And now I'm hoping that you can see my slides.
Yep, it looks great.
Awesome, right. So this is some work I did in collaboration with some brilliant biological oceanographers. So this was a very collaborative effort where we looked at trying to decode ecological complexity, so to speak, and using different methods, including machine learning, to determine eco-provinces in the global ocean. And in terms of oceanography here, it's looking at images like this. This is from the ECHO group. We can see that there are a lot of patterns here. This is surface currents, and the colors are temperature, and we can see that there's definitely something going on in the equator, and we've got these eddies spinning off the Agulhas region here, and things like the Gulf Stream sort of running along here. So there's clearly a lot of pattern, but actually determining those boundaries is hard. And this is where driving this intersection that you guys have been looking at potentially in this hack week of modern statistics, data science, and traditional modeling and theory work can really come in and create innovation, which is an area that I'm really passionately interested in. And in terms of this, novel tools and applications, I've called this a data science, which is a term that is very popular these days. And for me, the way I interpret it is shown in this plot to the right, where it's this overlap between geoscience or oceanography for us, mathematics, with a generous dose of statistics, and computer science, where you have things like machine learning definitely in there as a solid piece, but all of this sort of comes together. And the work I do is really focused on finding patterns, particularly also to be able to interpret the patterns that we may or may not find. And doing this, the work I'm presenting today, I'm kind of hoping might be a bit of a blueprint for what might be a good way of going about this, not just in biological oceanography, but also in other areas. For example, now I'm looking at an physics example from the subpolar North Atlantic. So there's really these methods can be applied basically across the board. And the presentation we just had really very nicely set me up, so thank you very much for that. Where the machine learning component here that I'll be talking to you about is mainly clustering, and this is kind of a particular branch of machine learning within the unsupervised component. And the thing that we're trying to do here is find these patterns. And the basic idea is that in 2D, us humans are very good at finding patterns, but if it becomes more complicated, say 11 dimensions, we become a lot worse at it. So here you see some different data distributions. There's some circles in the top here and some bananas in the middle, and we have lots of different data distributions, and the columns are the different methods that we can use. So they're different clustering methods. And the colors are the different clusters that you have found. And you can see that the different ones are better or worse at finding the different clusters. So here I've sort of semi-highlighted dbScan, which we'll be talking about later as well, and something like k-means that makes really strong assumptions about the underlying distribution of your data. And k-means is only, because of this, is only able to draw straight lines, while dbScan can do slightly more intricate things. So in terms of the trying to have a bit more of a technical angle to this talk, this is one of the things that that I would advise everyone to be very aware of in terms of the limitations. And I was also asked just to show where I found these different algorithms, and I thought I would do that basically just showing you. I really like scikit-learn. I think for most of the applications that I've been working with, they're really great. And here you can see a slightly more exhaustive list of different clustering algorithms with explanations. And these show you different use cases, and also for our applications that usually, at least in my case, have to deal with a lot of data. These little numbers down here tell you how scalable the algorithms are. So this type of information is very useful. And for dbScan, which we'll be going into, this is similar. You can find it on here using this very handy little map. So yeah, I recommend reading these things properly, and definitely also not just using the scikit-learn documentation to learn all this, but just using this as sort of a launching platform if you're interested in these types of methods. And with that, I thought I would stop briefly to see if any questions had come in.
No, I think nothing yet. Nothing so far.
Okay. Great. I'm assuming everyone is on board. So now we'll be going on to the science part of my talk, looking at trying to find these global ecological provinces, where the reason why we're interested in this is because they're very useful in terms of comparing locations for planning, conservation, and monitoring work, because you can isolate regions that are particularly sensitive, and also in terms of assessing the base of the food chain, for example. And for the terrestrial eco-provinces, these are well established. We are pretty sure about what's going on. On the right, you can see a map of what's called the Köppen classifications of the different climate regimes. But for biological oceanography, this becomes a little bit more tricky, because observational data is a lot sparser and more complicated to interpret, for example, genetics, and also that the ocean has liquid boundaries. So this is not a trivial problem, which is why machine learning approaches come in pretty handy. And what we're trying, the question we're trying to go after here is whether or not we can develop an objective method to classify ocean ecology. And one of the things that is very useful for this type of classification, if you're wanting it to be useful for things like conservation work, is that it should be useful both globally and for more regional work. So to really allow this interpretation or interpretability to be useful. So this is our objective going in. And the model that I was using for this, so this is based on a model, is a global biogeochemical ecosystem model that's a fully coupled physics, biogeochemistry, and ecology model that's building on a physical model called ECHO. And it's horrendously complicated. I mean, I'm not a biological oceanographer, so I mean, I'm coming at this from a physicist's point of view, but it has 51 different types with different what's called functional groups. So different phytoplankton do different things. For example, some of them are very good at making nitrogen available to the rest of the crowd, and some of them are really good at, say, not needing some of the other nutrients or not being eaten. And the green ones here are the ones that photosynthesize. They're the plants, so to speak. Then you have the grazers, called the zooplankton, and the ones that can sort of do both. And all the traits here are sort of set using observations. So this is a very complicated but a very good rendition of trait-based ecology, which makes it really interesting. But here I show you just 6 of the 51 different species, and we can see that there are clearly distinct patterns, but actually drawing the boundaries between them is hard. And for this machine learning application, I took the 51 different species and 4 different nutrients. So there was silicon, phosphorus, iron, and nitrogen. So we took the fluxes of these and the 51 different species, and this is what we started with. And the outcome here is something that I'll be using a little bit as the structure of this talk from now on. So first, we developed a way to look at and identify what we called eco-provinces. And this is a bit of a sketch of the workflow where first we used some dimensionality reduction and averaging with some different purposes in mind. We clustered on that and then projected it back. And then we developed what I call aggregated eco-provinces. So where we respected some of the limitations with our approach and used some graph theoretic sort of underlying logic and existing ecological sort of gold standards to arrive at what we called aggregated eco-provinces. This is the outline of the rest of the talk, and I guess brace yourself, there's some math coming, because the dimensionality reduction technique that made a lot of sense is called t-statistic neighborhood embedding. And I'm just going to dwell on this for a second, because one of the things that makes oceanographic data and oceanographic applications a little bit complicated is that there are a lot of assumptions that don't hold. For example, normality. If you're assuming your data is normally distributed, for example, when you're using PCA without using some sort of fancy kernel, you're making assumptions that can really skew what you're able to discover. Because, at least for this data, and I would argue for a lot of data in oceanography, the things you're interested in are those really fine scales, intricate, nonlinear interaction. These are really important to us and can really create insight. And one of the reasons why this t-statistic neighborhood embedding is great is because it does this dimensionality reduction, but it can handle the really complicated covariance spaces that we have in our data. So for this example, for every one of the different variables here, the different functional types and the nutrient fluxes, we have this 11-dimensional vector after some averaging that we have available for every Latin long. And these Latin longs now become our data points in this 11-dimensional space. So in this 11-dimensional space, we want to probabilistically project this down to a lower set. And here in this case, I used 3 dimensions. And the method that we use for this is called the Kullbach-Leibniz divergence. That's really a beautiful statistical inference. And sort of leveraging all of this, we can then do something that allows us to explore this data both in a way that makes clustering easier, although the t-statistic neighborhood embedding can sort of count as a clustering algorithm in itself. You know, we saw this complicated diagram in the talk before. But using this, we're able to do this very complicated data set justice. And just as an example of what this means, you can see here there's a pretty famous example of a test data set that's often used in machine learning called the MINUS data set. And here on the left hand, you can see all these sort of handwritten digits. It's a bit grainy, but here's a 0, there's a 4. So there are a lot of these, and the name of the game here is recognizing these different ones and using machine learning. And in the middle figure here, you can see that all the purple areas here are 4s, and they've been recognized. The 0s are sort of a streak here. But with PCA, the problem is that because you're assuming everything is normally distributed, things just sort of get parked on top of each other, because that's not true. And with TSNE here in this example, we're able to separate out the different classes much, much better. So this is the figure to the right. And this sort of more standard principal component analysis does much worse than the t-statistic neighborhood embedding. And these types of features, sort of the underlying statistical data, are definitely something to keep in mind and check using different methods. So just, I know that was probably really complicated. Please ask questions or talk to me or email me. Very happy to keep up the conversation. But in terms of just as a cartoon of what we're doing here now, we have this really complicated data. We flatten this using TSNE, so we reduce the dimensionality, and we end up with something like this. So this is our very complicated data space, now reduced into 2D just for visualization. And each and every one of these different dots is one lat long. And having this now, this might look like it's not really helpful at all. But if we look at a hex plot or look at how dense the different regions are in this data, you can see that there are some that are much denser than others, and they stand out. So doing this now, you can imagine me looking at this and then going back to that scikit-learn table with all the different clustering algorithms and going like, the density-based one, that's what we shall use. And this is basically what happened. I'm simplifying a little bit. But doing this, I selected the density-based spatial clustering of applications with noise, which is a short and snappy title that boils down to dbScan. So dbScan is what people call this. And what this does is it takes the data points here in different colors, and it sort of stochastically just starts somewhere in the data set. And then it looks at how many points are around it within this distance, this epsilon distance. So here, if you imagine us starting here, we draw this curve around us, and we see that, okay, these points are within my radius. And then it starts to crawl along the data. So it finds this one is on the edge, and this one is still within it, so it adds this to the cluster. But then this is the only one that's within this radius, so this then becomes the edge of that cluster. And you set the minimum number of points that you want to be able to call something a cluster. And if you end up outside of this, then you're classified as noise. And this ability to classify noise is actually also pretty handy, because sometimes, with oceanography, we have pretty noisy data. So this is the algorithm that I found to be useful. And doing this as well, being able to be cognizant about how you set these parameters is also pretty important. So here you see a few graphs that I plotted to have a look at this, where I varied the number of the minimum points I could choose and the distance epsilon. And what you want to do is find what's called this elbow point, where things suddenly start to increase a lot in terms of how connected the data is. So I selected this point over here. And doing these types of things is also really important. But having done this now, we finally get to the interesting science, which at the end of the day, is what I care about. So having done this dimensionality reduction step and being able to visualize the data and see what's appropriate and clustered on it, this ball of data that I've affectionately called the yarn ball is the product. And we can see that there are really interesting, intricate structures. And with dbScan, we can really sort of get at those, which is great. And remembering that each and every one of these different dots is a Latin long, we can then go back and project it onto the globe, which is what this looks like. And seeing this, having clustered the nutrients, phytoplankton, and zooplankton, we see things that we expect to see. So having different sort of sanity checks, both statistical and otherwise, is a really great way of sanity checking in as many ways as you can. And I'm not a biological oceanographer, so for this, I asked my friends and collaborators, But looking at this, we can see that there's a lot of zonality, say, in the Southern Ocean here, which is great. That's what we expect. There's also something distinct going on in the equator and something going on in these regions, the wind gyres or the oligotrophic regions, if you will. So looking at this, it already looks really promising, which is a very encouraging platform to move on from, where I now segue into the second part, this aggregation step. So trying to arrive at these aggregated eco-provinces, because in the full space we arrived at order of 100 different ones, and this is, in terms of getting a global picture, a little bit overwhelming, at least for me. So being able to aggregate them is really useful. And in terms of also trying to make it useful, we looked at what's currently still the gold standard for this type of classification of the different ecosystems in the ocean, which came from an expert in this type of oceanography, Longhurst. He published this in 1995 originally. And this was sort of a synthesis of his expertise and available data at the time. And you can see that there's a lot of overlap between the full eco-province system that we had and what he drew out. But his boundaries are very static and also, somewhat straight, which seems counterintuitive for an oceanic system. But these are very useful, these Longhurst provinces, and they sort of form the benchmark that we'll use for the rest of the talk.
Maike,
And yes.
before you dig into this section, there are a few questions that have come up, and I think
Wonderful.
they're relevant to what you've just said.
Mm-hmm.
So, yeah, I hope you don't mind me interrupting you, but you mentioned you
No,
didn't.
not at all, not at all. Please do.
So, let me just read a couple of these. So, I remember reading that TSNE isn't great for classification problems because it depends on the entire data set. So objects from the same class, but a different set, may not map to the same cluster. Can you speak on that?
Sure. So TSNE has a lot of different parameters you can play around with, and one of them is the perplexity, so to speak. So one of them kind of lets you set to what extent a local area is forced to care about the global area, or some variation within that. And this is actually, I'm really glad you asked this question, because this is kind of a point in this sort of very sort of delicate process of trying to find these types of robust eco-provinces. So I'm hoping to actually address this issue of stochasticity and the issue of arriving at something robust a little bit later, because it's a really, really great and actually somewhat profound question that also digs into the technicalities of the TSNE method itself. But in terms of getting things in the wrong clusters, so to speak, this is something that you can fine-tune using this method. And also, very importantly, you can, with your understanding of the data, sanity check it a second time. So, for example, if you were to use something like PCA and then throw something like k-means on it afterwards, this, and I checked, will fail the statistical tests you throw at it. So, in this sense, you have a rationale and a reason to make things more complicated. And with TSNE and with this series of sanity checks that I'm hoping to get to, you can sort of get around this. But the short answer to your question is that there's a parameter called the perplexity that you can set to make sure that that you avoid that problem. But that's a great question.
Okay, just a couple more. Thanks for that.
I'll try to keep it more brief.
Oh, no, that's okay. It looks like these ocean climate types being clustered into lat-long regions. Are you also thinking about depths and or stratification layers by temperature or oxygen or light or ... How do these regions compare with water masses and or water types?
That's there are a lot of questions in there. I guess the short answer is no. So we just took the 20-year mean of the surface biomass present in the different types or the different species or what have you in the Darwin model. What I would have loved to do is to go down to, say, the bottom of the euphotic layer to also be able to get at things like deep chlorophyll maxima. But for computational reasons, and the fact that, this is the first time we've tried something like this, we weren't able to do these types of things. In terms of the second part of your question, for the sort of the physics comparison, this is how I interpreted your question, but please correct me. This was something that we did, but only by eye. I have another paper that was published last year looking at different physical oceanography dynamical regimes. And within that, I'd drawn some, or discovered some regimes within global oceanography. And we compared those to the different AEPs, or aggregated eco-provinces, that we found in this one. So it's explicitly no, but sort of by eye, yes. But I think there's huge scope for doing that type of thing. So that's a great question. Maybe in the interest of time, I'll jump to the next question if there is one.
Yes. What do you think about the asymmetry of KL to measure distances among low and high dimension? Can that generate some bias to go back and forth?
Potentially, yeah. That's actually also a great point. I was wanting to touch on the methods that you use to do this minimization or this gradient descent that the Kurbach-Leibniz divergence uses. So in I think using this divergence is definitely the best way forwards. But of course, you can introduce errors, and being aware of this, I think, is very important. And this again comes back to this consideration of what your methods are introducing, and the things that you probably want to be aware of. So I'm trying to keep it brief.
Yeah, no, that's great. One last one, which was mine. I often do an eye test like you described to see how I'm doing, but I sometimes worry that I'm introducing my own kind of expert bias. Do you feel that way when you do those kind of sort of gut checks? Do you worry about that?
Yes, to some extent. I mean, I think you can only do as well as you can. And in my experience, if you say use k-means that has strong assumptions on something and it looks great, you know that there are things you can do to check whether or not what at the end of the day is a statistical model you put on your data is doing well. So with something like k-means or a more classical, I guess, regression problem at the end of the day, you can use what's called information criteria. And knowing about this and putting those on and doing that check, in that sense, you're not just left with that looks pretty good. You also have some more formal way or a stronger rationale for saying, I think this is good. But at the end of the day, you can only disprove a hypothesis. You can't prove it. So even within these machine learning applications, you're still sort of stuck with that at the end of the day. And one thing I think it's very important to note with these types of applications, kind of in contrast to the luxury we have in physics where you can derive things, is that I selected these variables together with an ecologist, or a biological oceanographer. And in that sense, I've already probably introduced a lot of bias by choosing those variables. So maybe there was another variable that was really important to explain this. I might be blissfully unaware of that. So in this sense, that I think would be the largest area of bias. But also by being able to choose the variables and sort of set up this, this problem, you're also setting yourself up for interpretability. So in this sense, because we added in everything we thought we would need to paint an understandable and sort of tractable picture of what these provinces would be doing after we, discovered them, we tried to bake in this interpretability. So, so yes, bias is definitely something to be aware of, but I think it's sort of a double-edged sword in some ways, because at the end of the day, these are tools and you have to use them, but they're not perfect. Even just choosing the tools you're applying, you're introducing some form of bias.
Thanks. Those are great insights. I think that's all the questions that are up there now. So,
okay, great. Well, please stop me. I'm just talking to my screen right now, so having conversations is much nicer.
There's a lot of back and forth going on too, and folks are answering the questions on the fly. So you're definitely generating lots of chat.
Great. Wonderful. Okay, shall I press on?
Yes.
Great. Okay. So now we've come to this aggregation component where we take a little sidestep from just machine learning and move into sort of a more sort of what I would call graph theory application, where we have these over 100 different eco-provinces, and in terms of interpretability, for me that's too much. But a metric that's been used in ecology for a really long time is something that can come in really handy. So here we looked at what's called the Bray-Curtis dissimilarity, and this is a metric to say how similar one species composition is to another one. So in this sense, we can compare every single eco-province to every single other eco-province and get one number for how similar they are. So here you see the equation. It's effectively the different types that are present divided by the total types that are the same. So here for one eco-province, if it's completely similar, it's black. So if it is itself, then it's black, and you can sort of see it here. And if it's completely dissimilar, so they're very, very little to do with each other ecologically, you're sort of more yellow or white. So in this sense, we can see, what it's doing. It's sort of selecting these similar areas. This is what you can see. So this is effectively one row or one column here. This is the graph is symmetric. So having this is really useful because we can then go on and do something that's a little bit like what Facebook does. So here you have just the structure drawn by who knows who, and in this sense, we're trying to do this where we're trying to sort of construct a graph consisting of a set of points and the links between them. And here, if you see this guy, he knows all of these people and is sort of familiar with all of these people, and directly with this guy. But it's it's a very complicated picture, but it's something that allows us to draw similarities, because all of these are so similar to this one and sort of relate just to this one. So in terms of the Bray-Curtis dissimilarity, where we now have one number to represent these links, what we can do is draw these links. And here I've arranged the different provinces around here in a ring. And we can see that some of them are really popular. And if you, I just sort of inverted the color scale here for visual intuition, where you can see that if something is very bright, then a lot of things are very similar to this. So this eco-province is basically like that very popular guy in the other graph. And having this, and knowing that everyone is connected to everyone else, we can then start to sort. And this is what this aggregation does in terms of the aggregated eco-provinces. So if we see that, okay, to make a useful global analysis, maybe we want 4 different, mega eco-provinces. And this means that we can take the 4 most popular eco-provinces and sort all the other eco-provinces into the one that they're most similar to, which allows us to take these 100+ eco-provinces and just sort of collapse them into 4. And doing so is great, but I mentioned this problem of stochasticity and the fact that we have this gold standard. We have this Longhurst map that shows us, what people have been using so far and what's been really useful. And doing this again, you go back to ecology and go back to your sanity checks and say, okay, to be useful, we probably have to do better than Longhurst, but how do you define better? What is better? And doing this, we again used the Bray-Curtis dissimilarity, but asked not what the average of those different eco-provinces, where we asked how self-similar the different components of the eco-provinces were. So how similar is one lat long in terms of all the other lat longs within that eco-province or aggregated eco-province. And here you see how self-similar the Longhurst map is here, and the colors are the same here. And this is what this green line is, this benchmark. So we want to beat Longhurst, we want to be below Longhurst, but you can notice that there's some error bars here. So these are a representation of what the different aggregation levels do. So how many sort of very popular eco-provinces we choose, and the error bars here are what I was hoping to spend a little bit of time on. And this comes from the fact that we have some stochasticity baked into our algorithm, so the way we've chosen to set this up. And the reason why this is interesting, and the reason why considering things like how you do, or how you sort of need to consider these gradients that you're walking down is what comes in here. So with this, this is again scikit-learn. Scikit-learn is great. With this TSNE algorithm, if you sort of read this, there's a lot of information in here. And here, for example, we can see that the default is Barnes Hut, and this is how this minimization of your projection from the very high dimensional space down to the lower dimensional space happens. And this might seem very technical and dry, but it's actually kind of important in the sense that in 2D, now remember we started in 11-dimensional space, so in 2D we can see what happens. In 11D, at least for me, that's difficult. And in this space, so to speak, if we think of something that's good, we want to be able to arrive at one of these tops here. So this would be our goal. But if you imagine starting this, so having this algorithm being somewhat stochastic, so using TSNE, you might start, say, at this point, and this minimization using these different algorithms or methods would then, if you end up here, sort of climb up to this top. And with this, I mean, this is quite smooth. This works quite well. But if you because of the stochasticity, started here, for example, you might only find this little local minima. And ideally, what you want to find is this global minima, but with the realization that that's probably really, really difficult for these applications. Taking this into account is important. And this is what we're trying to do with this step. So in this sense, the error bars here are to do with the fact that there is this stochasticity. And the reason why choosing this aggregation level, together with the fact that it was better than this Longhurst, this previous province identification methods, was also that the error bars are they're narrower. So this means that if you repeat the entire process, you're within this error bar for getting the same provinces. And because this standard deviation sort of increases further out, 12 is the lowest recommended aggregation level that we set, because you are below this, and it's more useful than longer. So there you go. But it is really, in terms of designing these types of experiments, being cognizant of these things is quite important. So also cognizant of the time. I will press on. But in terms of the utility also, I mean, this might seem very dry and boring, but just in terms of what you can use this for, then it actually becomes a major benefit, because if you see these, so these are the different aggregation levels here, where the degree of aggregation or nesting increases as we move to the left. You can start with something that might be pretty relevant for a coastal application if you are interested in that area, which, if you're interested in fisheries, that's probably potentially a great level, although the model doesn't do so well around the coasts, but yeah, what have you. But LMEs are what's called a large marine ecosystem that are also used pretty widely. And if you're interested in these and how this model does in comparison to these, maybe an aggregation level of 20 is better. But for these global applications, the minimum is 12. So this is a way in which this can actually sort of be used to our advantage, and makes it so that we can address this nesting that is actually desirable for these types of eco-provinces. And for my own simplicity, we also then looked at the global picture here, and this complexity or aggregation level of 12. And doing this now, we can go back to what is present in terms of the interpretability here, what's present in these different aggregated eco-provinces. And here you can see in the bottom here, we've labeled them A to L, and they correspond to these different regions. Here we see A to L again. And on the y-axis here is the biomass. And one thing that was really interesting to see was that by doing this classification, we can learn things that you wouldn't be able to see if you were just using bulk chlorophyll, which we get from satellites. Because this level of biomass here for the column that's more colorful on the left side of the brown column is a zooplankton. If we compare this phytoplankton column here to another phytoplankton column here or here, we can see that although they would have pretty similar chlorophyll to be observed, their composition is actually really different. But also, very interestingly, this column and this column are actually ecologically very similar, although they would look very different from a just bulk chlorophyll standpoint. And in addition to that, also, in terms of predicting where there might be a lot of zooplankton, which you care about if you're interested in the higher trophic levels, for example, fish, there is this trophic amplification that biologists are very aware of. But in terms of seeing where this might happen or where it might not happen, knowing more about this ecological composition is important. And here you can see that, for example, in these cases, although there's a lot of biomass, this doesn't really get down to the fish population in terms of just how much zooplankton there is. But if you're in this area, there's a lot. So this composition can clearly support more of this type of production. So in this sense, just this sort of global aggregation level at at 12 aggregated eco-provinces can already start to be something that can tell a pretty interesting ecological story. But I should stress here, I'm not a biological oceanographer, so this was a very collaborative effort. But the model is also, a cartoon of a real ecosystem. And an important step for this also in terms of sanity checking the utility or the usefulness of these types of approaches is combining this with in situ data and in situ observations. So this is a step that's still outstanding, but something that I would be really keen to collaborate on, just to move these types of efforts into something that's actually useful. So with this, I was going to summarize with that we, using a combination of different methods from machine learning and statistics and graph theory, objectively uncovered ecosystems in the global ocean, and this aggregation of these global and regional eco-provinces towards different applications. And the methods we used were this probabilistic TSNE and DB-scan, together with the aggregation using this dissimilarity metric. And we saw that if you have similar chlorophyll, you might have very different ecology, and vice versa. And we also saw that this ecological impact could have a radiating effect onto zooplankton abundance that we can already see using just this 12 AEP aggregation method. And if you're interested in these different aggregation levels, there's a little plotting tool on my GitHub. With that, I had a thank you slide, but that seems to not have made it. So I'll just leave this here. And yeah, thank you very much for your attention. I'm hoping there might be some more questions for me.
Yeah, that was great. Thank you so much, Maike.
No worries.
There's definitely some more questions in the Slack channel, so I'll just kind of start with some of these. I think there were some answers, but I think it would be great to have your answer as well. Has all of this been plotted and programmed in Python?
Yes. A lot of it came from scikit-learn. Some things I wrote myself in terms of the Bray-Curtis implementation, but all of it was in Python. I strongly support Python, especially with the different efforts related to the Pangeo project and just this zoo of different really wonderful efforts just to make these things more accessible. Obviously with the double-edged that because they're more accessible, it's easier to make mistakes, but I think that learning curve is probably well worth it. So yes, it was done in Python. Yeah, also a lot of this happened on HPC facilities where something like Anaconda available, where I could just sort of plonk my environment down somewhere and basically use it out of the box, was incredibly useful. Again, I'm keeping this somewhat technical, so please let me know if I'm going overboard.
Oh no, I think that's great. Yeah, no, thank you for that explanation. Another question that came up is, what additional data sets would be most useful, in your opinion, to help better differentiate VGC provinces?
Great question. I guess that would depend entirely on what you want to use it for. So biogeochemical provinces, of course, can kind of mean a lot of things. So I've been, together with some other folks, thinking a lot about whether or not we could define these more in terms of the tropodynamics that are going on, so explicitly get at these energy fluxes, which in terms of targeting, for example, a fisheries application, could be great, where you would need the tropodynamic variables, but then also the fisheries data sets to validate these different methods. one thing to note is that all these different province identifications haven't really been systematically compared, and this is something that that I think is an outstanding issue that, you know, potentially for the next hackathon you could you could think about, because I think there are a lot of ways of doing this. In terms of, say, more of a in situ data collection tool, I think data sets that also could be useful are things like the ones collected by the Gradients effort that's also going on at UW, where every year they sail, they collect data from Hawaii going north, and using this data set I think could also be very interesting in terms of matching up where you're expecting to see something and where you might actually see it. Of course, satellite data is also hugely useful, and potentially also with the new effort, I've kind of lost track of what's happening with it, is the PACE satellite, I believe, that's going to be able to look at much more finely the different wave bands. And I mean, there are probably biological oceanographers in the audience here, so please bear with me. But knowing more about the different wave bands, you could say a little bit more about what the species composition that might be present is. So I think there are a lot of different data sets that might play into different applications one might have in mind.
Great, thanks. Another question that was sort of more of a broad question is ocean science keeping up with machine learning advances? If not, where are we lagging?
That's a great question. I mean, I think that's maybe where someone like me with a background in what, when I was doing my PhD, was called complex systems, which today might be branded AI or data science, might have a bit of an edge. It's in terms of insight there. So I would say oceanography has a little bit left to learn. I think also the methods that are being used in core machine learning might not necessarily be optimal for us yet. And I mentioned the issue that a lot of the methods that might be routinely applicable to machine learning problems, like employing PCA straight out of the box, might actually be at our disadvantage, because if you use PCA, you're effectively assuming that your data is normally distributed and sort of disallowing yourself to really hone in on those very interesting nonlinearities that we often have. And if you've already sort of accidentally normalized your data, then something like k-means that also has this underlying normality assumption can come in and actually do a pretty good job, but leave out what the thing was that you were actually interested in to start with. So in that sense, I think the tools are often things we're not entirely aware of, like these different aspects of dimensionality reduction. And they're also evolving very quickly. So now you have one that also uses manifolds, which I'm very interested in, rather than just as probabilistic projection, also for scalability. And together with this danger of not entirely knowing what the implications are of using these methods straight out of the box for us, because our data is different and our objectives can be different. And this communication between these different fields, which effectively it is, I mean, most of these efforts are just inherently very interdisciplinary, and communicating these things is sometimes very hard. So I think there's a lot to learn on both ends, really. So that was a very long-winded answer.
No, that was a great answer.